I am making you a video, and I hope you can hear it, and it doesn't just sound like the fans of my laptop, because they're really fast right now, which seems unnecessary, but it's probably because I've been dancing yesterday. So that is going to help you make these vlogs. I'm just going to anything terrifying to look at. So what we're going to do is look at my lecture from this morning, which is obviously once you're there, I am just going to talk to you about how I'm going to be done for the entire rest of the day, and potentially watch first class, maybe first class, this which first class, or maybe first class, just actually I watched my lecture last week as well. So maybe we'll do that. Wow, the chair's pretty creaky. Never noticed that before. So, oh, there's a little creature. It's like a, I think it was Jabba. Yeah. But it's not. It's called a donut. I'm just gonna look it up. Donuts are very narrow-footed marsupials the size of a mouse. Members of the genus Smithopsis. They are mainly insectivorous, which means they are exceptionally cute, and they look like birds. Maybe we'll watch them. I think I've seen them in a David Attenborough documentary. It might have been Life, actually. They've got the legs. They're exceptionally cute. They don't look anything like a giraffe, but they do look very cute. They haven't got ears or tail. They've got a tail, but they've got legs. They're exceptionally cute. Okay. I've just accidentally started watching Quicksilver videos, which not Quicksilver, but Evan Peters videos. I felt like I had to look up. It's just too cute for the world. I don't know how anyone says how someone doing this. Okay. So I'm not excited. I'm not actually talking about this. But it was exciting. And it was really good. Like, the last bit of it, I was like, if Hank had the technology we have now back then, he would be so excited. Oh my god. Like, the sequencing that we've been able to do in terms of sequencing the genome has been incredible, and learning about how genetics affects evolution and okay, so last week we learned about variety of life, which encompasses variation among species and other biological elements, including alleles, gene complexes, populations, communities, ecosystems, landscapes, and biogeographical regions. I can't remember this well enough to tell you. It's only a week ago. I just have no brain power. I love the notes. This lecturer I have for biodiversity is brilliant. His name is Andrew. Professor. But I'm writing stuff down, and I just have these random things written in the margins. So from last week, I've just got talking about penicillin and Linnaeus, and then taxonomy and how things are named according to species, genus, family, order, class, phylum, kingdom. And then randomly at the bottom, randomly at the side, I've got pigeons can smell and swallow. And down here I've got go bird watching. Like, why do I have this? And this week is the same. I've got the stuff we were doing, and then just randomly at the bottom I've got Amazon is wide at its mouth as the Thames is long, and the Thames was a tributary of the Rhine back in the last, way back when, not very far, the last glacial ice age. Back in the last ice age, when the sea level was lower and the masses were interlinked by bridges and stuff. So England was linked to the Netherlands. And the way the Thames flowed, it was actually a tributary of the Rhine, because the Rhine came in the Doggerlands instead of in and then here I've got a page on biological species concept, which we wrote yesterday and which apparently is wrong, which is fine. And at the bottom of the page I've just written Ice Age, Exmoor but not Dartmoor, oceanic influence despite height, which is basically in the last ice age north of England. Something else was ice, and everything below was tundra. But Exmoor was glacial, but Dartmoor wasn't, even though Dartmoor is higher up. So the higher altitude means colder, but it's actually the fact that it's close to the sea, so the oceanic influence meant that it was a state of I don't know about physics to know what that really how it works. I know the sea is warmer. That's what I know. We were talking about plants, and it was really interesting. I'll tell you about it. But I've just got written down. All bread is hexaploid wheat. Something else I've just randomly written down. Apart from done art, I wrote that down specifically to talk to you about because it was really cute. I just don't really know much about it, just because it's cute. Okay, so I'm going to go through this lecture, which was like 3 hours of lecture, so obviously I'm not going to take that long because some stuff you'll know or you'll understand really. Okay, so we were talking about the origins of species, not the origin of species, but the origins of species with an S on the end. There are more than one way in which species can originate. Or a lot of when Darwin published The Origin of Species, which isn't actually its name, it's got a really long name that nobody can really say, so it's just called The Origin of Species. Lots of people were like, well, if you didn't actually, within the book, define how species formed, or even though it's called The Origin of Species, you didn't actually talk about how species formed. So when those publishers were reading it, they were aware of the missing link. There should be missing links between species, between what we had and what we used to have, what we've got fossils of, and what there is now. And then, fortunately, that was when, almost exactly the same time, people discovered Archaeopteryx with fossils, fossilization. Can't word fossils. And And that was like exactly what we were talking about in terms of having something in between. So Archaeopteryx is between dinosaurs and birds, I think. So yes. Anyway, that's a missing link. Like, oh my god, this really works. Appreciation. But defining a species is very difficult and it's quite subjective depending on who you ask. And nobody's wrong necessarily because there isn't a rigid set of definitions, because the way we tell what species is, the way we compare species changes all the time. So yeah, it's not exactly what we thought it was or what we think it is. For example, 40-ish years ago, if you were asked to say how many great apes there were, the likelihood is they would say there were 3, and the 3 would be the chimpanzee, the orangutan, and the gorilla, if we exclude humans. But now, if you ask, people are very likely to say that there are 6 or 7, in fact. And that's because people discovered that there wasn't just the chimpanzee. There was the chimpanzee and the bonobo, which are different. They are physiologically very similar, behaviourally quite similar in a lot of ways, but they are actually distinct species. And with the orangutans, you've got the Sumatran orangutan and the Bornean orangutan, and with the gorilla, you've got the Western gorilla and the Eastern gorilla. And the Eastern gorilla is then split into the mountain gorilla and the lowland gorilla. Eastern mountain gorilla, Eastern lowland gorilla. And the speciation, there are lots of different ways in which we survive, but this one is based around natural barriers. So, we didn't actually talk about the birds, we talked about chimpanzee and bonobos, being that they are split by the Congo River, which is huge. It's one of the 3 largest rivers in the world. No, it's not longer than the Nile, but it contains huge amounts more water than the Nile. It's got like 1,000 times more water in it. It's huge. And so the chimpanzee spreads from the Congo all the way up to Senegal, almost, up on the west coast of Africa. But the chimpanzee spread is north of the Congo River, whereas the bonobo is very specifically on the south. And this is my diagram. It's a really great diagram. That's the Congo. I can't do this backwards. That's the Congo. And then the X is the bonobo. And that's where they're split. They're split there, and it's a huge natural geographical barrier. So chimpanzees are on one side and bonobos on the other. And in terms of the orangutans, whilst again they are quite similar, they are in lots of ways very different. And the Sumatra and Borneo were last linked in the last ice age, which is about 10,000 years ago, because it's in the South China Sea, which is very shallow, and as I said, things are glacial. Land is just like in the North Sea, it's all very shallow. So potentially, what was surprising is that there's only 10,000 years since they were last linked, but the variation is considered that there should be, it suggests that they'd had a longer separated. And so then people looked at DNA. Yay, DNA! And the DNA suggested that they've been separated for more like 1.4 million years. which is interesting, but also makes a lot of sense given that they don't migrate or anything. They're very kind of sedentary. They stick within their habitat and they don't really move anymore, go anywhere. The interesting thing about the orangutans is when they're kept in captivity in zoos and stuff, they weren't kept together. They weren't kept as separate species. They weren't kept as a Sumatran Mongolian. They were just kept as orangutans, even though they're quite different to look at in a lot of ways. And they hybridize and they produce fertile offspring, but whether they are two separate species or whether they are subspecies is a big question. And the question comes down to, are they on different evolutionary paths? But is this a splitting? Are they at a splitting point? Are they distinctly on different evolutionary trajectories? Which is where we came to the biological species concept, which is really interesting because this is what we were talking about yesterday. We were talking about Eric and getting it wrong about mutants being a different species, and how I said, as my evidence, that species is defined by whether, or are partly defined by whether they can reproduce to produce fertile offspring. And it's wrong, basically. The idea is just, in some ways, it makes a lot of sense. And it was brilliant when it was conceptualized in the 1930s. It was brilliant because it was kind of the first time that evolutionary biologists had looked at defining a species by something conceptual rather than just as something physical. So rather than looking at things and going, oh, this is a physiological difference, and this is this physiological difference, and this is behavioural difference, this is this. So therefore they might be X and Y related, or this related, or whatever. Somebody actually thought of it in a different way. So it was very exciting theory. Except it doesn't work, because it doesn't work with asexual species mammals, it doesn't work with bacteria or unicellular organisms, and it doesn't work at all with plants. So it doesn't work. If you want to use the same definition of species for everything alive, it doesn't work. So we moved on to looking at other things, which is very exciting, actually. I'm just adjusting the lamp. So we look at we still look at morphological differences, because does it look similar? Does it look completely fucking different? We look at differences in DNA, which is actually, we looked at that more at the end of the lecture, so I will talk to you in more detail about that, but not it's Carrie. It's really actually quite interesting. And we look at whether species are on different evolutionary trajectories, so if they are going in different directions. So the line between races and species is very blurred. This was an interesting part of our lecture last week, was about people used to use race all the time to mean subspecies, and now because of the political connotations it doesn't get used as much, but yeah. So the line between races and species is very blurred, and often follows the path not of anything particularly scientific but of fashion. So in Victorian times everybody was like, everything's a different species, they're all species, everything is a species. And so they separated everything was a species. Then in the 20th century, scientists kind of took a different approach and went, we've got to lump everything together. Which is kind of how subspecies were always born, was people going, oh, they're not different species, they're just subspecies, the same thing. Yeah. And now, 21st century, a lot of things are being separated again, which is in part by the fact that we've got DNA sequencing, but lots of subspecies are split into species, and there's a political motivation behind that as well, which isn't a bad thing. Sorry, I'm back. You're back. Which is just that, right. The political motivation is in terms of conservation. It's easier and more effective to talk about conservation of a species than it is of a race. So you want to say, save the orangutan, say the Bornean orangutan is doing fine, the Sumatran orangutan is doing terribly. If you separate them into different species, you can then talk about the Sumatran orangutan. The orangutan is endangered. This is endangered, rather than if somebody just thinks it's a subspecies, they go, oh, there's loads of rhinos, loads of this, loads of that. What do I care about this subspecies? So if you make it about species instead, then yeah, there's a lot of separation that's happening, which is not so bad thing. If it increases conservation, it's probably a good thing. So we talked about natural barriers causing speciation. What we've also got is isolation. Hey, beautiful ones. So we were talking about the white-throat, which is a little bird. Obviously, kind of endemic to Britain, plus the USA. But there is stuff later, plant stuff, which is really interesting. So we're talking about the white-throat, and we've got the white-throat, and we've got the lesser white-throat. And they are two entirely distinct species, even though they look very similar. They've clearly come from the same place, common ancestor, and so they both come here in the summer. They have some visitors, they migrate, and but the white-throat, common white-throat, migrates from Western Africa. So it likes the savannah, Nigeria, Burkina Faso, Western Africa. That's where it migrates from. And the lesser white-throat is very similar in appearance, and it's also migratory and comes here in the summer. But it migrates from Eastern Africa, and it's ever so funny because it comes a very long way around. So the white-throat, common white-throat, migrates from Western Africa, and it comes straight across the Straits of Gibraltar, over Spain, and up to us. Whereas the lesser white-throat, migrating from Eastern Africa, goes all the way around via Israel and Saudi Arabia, and then comes across Europe to us. Now, they are different species. They have different songs. Now they moved again. They became isolated from each other again because of the glaciers. Yay! Love ice ages. They do incredible things to evolution. So basically the bird couldn't live in the UK. It was too cold. So the white-throats from the western Africa, instead of migrating further north, migrated to the Iberian Peninsula. So they were in Spain and Portugal, which had, during the Ice Ages, southern Spain and Portugal was the same sort of climate as we have here now. The lesser white-throat migrated from the east of Africa, and it migrated up to Greece and stayed there. Again, because it was too cold further north. When the glaciers retreated, both sets of species which had come from two different places, when the glaciers retreated, they went further north again. And central, so they moved centrally into more central Europe, and they moved further north again, and ended up back in the UK. And now, when two species like that come together, in that situation, they either intermingle, or they diverge. And there were several things that cause divergence, particularly reproductive barriers. So I'm not really entirely sure why. There's a number of reasons. Reproductive barriers being their songs differentiated. So their songs are turned into different. Their songs are developed and changed. And the suggestion is that if they had hybrids, and they intermingled and they interbred, even if they did, which is less likely because the songs were different, but if they did, the hybrids wouldn't have necessarily survived because they would have had a different song, or they wouldn't have known which song to sing. They wouldn't have known which way to migrate, whether they were supposed to migrate eastwards or westwards, and they would have just got lost. Potentially just got lost. Which is very sad, really. But that's what's happened, is that they have then separated. So they are glacial species, which means they've diverged within the last 2 million years. Because of repeat glaciation, this happens with lots of species, but repeat glaciation has meant that that's thick, it's just growing and growing, and the western, the common white throat is actually split as well into western and eastern places. That's obvious. But it was really interesting. Oh my god, glaciers! They just shake everything up, fucking everyone over. So then we looked at the carrion crow and the hooded crow. Again, this is European, so I don't know how helpful this is in terms of, but you can look them up. I'm sure it's amazing. The fascinating thing, I have to find your map of where they sit in their distribution, basically. But they exist in opposite places. So the carrion crow is resident in the UK, from all the way from the very south and the Isle of Wight all the way up to a very distinct line across the whole of Scotland. And on the other half of the other side of that line, from the very top of Scotland, is the hooded crow, and the entirety of Ireland is the hooded crow as well. But there's no carrion crows in Ireland, and there's no carrion crows in the northern bit of Scotland. And it's actually their distribution follows a line, a very distinct line across and around throughout Europe. You can find it better. Yeah. Kind of like across Norway and the tip of Sweden. Or include Sweden and the tip of Norway, something like that. But the interesting thing is there's no geographical barrier. So they're not separated by anything that would force them to be different. There's no glaciation. They're not migratory. And when they hybridize and they produce fertile offspring. So the question is, why does the hybridization not increase? So the space in which they both intermingle and breed and produce fertile offspring, and you get these crows that, they've got look like hooded crows, but they're varying degrees of grey, so less whitish and more darker. That's bad English. So this is where you find that you have a balance, where on the edges where they do intermingle, they do breed and they do produce these offspring. But there is significant evidence that shows that their offspring are less successful, and they produce fewer successful offspring each time. So the birds that are hybrids are somehow, not sure how or why, but they are at enough of a disadvantage that they don't increase. The number of them doesn't increase, but they're not at enough of a disadvantage that they don't breed together at all. So there are hybridizations, they're just not very common, and it doesn't grow because they are somehow at enough of a disadvantage that does happen. It's pretty cool. Then we looked at gulls, and again I'm going to find some pictures. but or you could look them up. Either way. So we talked about the herring gulls and the lesser black-backed gulls, and you see them in England as two very distinct species. The herring gull is one of the most common ones. It's got a very pale back, and it's got very hold on. It's just leg colour. I just had a moment of madness. Yeah, okay. Okay. So the herring gull is very pale back, like a gull, basically. They haven't got big angry beaks like gannets, but yeah. Dark tail, pale back, pink legs. Then you've got the lesser black-backed gull, which has a very dark back. They look quite similar, but it's very dark-backed, and it's got yellow legs, distinctly yellow legs. And the fascinating thing is that they are completely in the UK. They are completely different species. Very distinct differences, different migratory patterns, different behaviours. They breed in different ways. The herring gull breeds in cliffs. The lesser black-backed gull breeds on flats, on islands. So the Scilly Isles, for example, they breed on the flat. In the cliffs, they breed on the flat. And so they are different species. But what's really fascinating is if you go, if you look at the lesser black-backed gull, they breed together, and you get these gulls that have paler backs or darker backs, and pinkier or yellowier legs, somewhere in between the two. But if you go around the world very slowly, you discover, and you take a look at the lesser black-backed gull. If you head into Eastern Europe, you have something that's it's got a bit of a pale back. Its legs are a little bit less yellow, and if you go around a little bit more to Asia, you get a different kind of gull, which is the same. It's definitely the same gull, but it's, again, the back's a bit paler, the legs are a little bit, less distinctly yellow. And if you keep going around and around and around, you end up in the USA, and you end up with a herring gull, which looks, it's still the lesser black-backed gull, and it looks like the herring gull, and it's got the pale back and the pink legs, but if you've gone around the world the right way, it's such a gradual shift. It's almost impossible to differentiate them as different species until you get to Western Europe. And what's happened in Western Europe is the herring gull crossed the Atlantic. It just decided in the 1800s that it was going to cross the Atlantic, and you had a bunch of herring gulls came over from the USA. So what was actually a really gradual shift in its physiology and behaviour around the globe the right way actually ended up, once it crossed the Atlantic, you suddenly end up with these two very distinct species, and they are very distinct, but if you go around the world you can see the shift, and it's so gradual you can't differentiate them as different species until you suddenly get them both together in the same place. And there's all these intermediaries. Yeah, it's crazy. But amazing. Okay, then we've got making them again. So there's different kinds of separation. Geographical separation, things that we use to define, again, to tell whether things are different species are the same. Geographical separation, ecological separation, physiological separation. God. Do you want to hear about polyploidy? So you know that I don't know, I assume maybe human cells have 23 sets of chromosomes in them, 23 pairs. And that insect cells, that gets doubled, and then half, and then half, and then you end up with a person with the right number. And our cells are called diploid cells because two sets, and each chromosome has a pair of some kind that it matches up with. Now, plants are very clever, and what happens often with hybridization is the reason you get things like infertile offspring and such is because when the egg and sperm cells combine, a scientific term, so it's less mammalian, but you get the chromosomes don't line up, don't always line up. So what plant cells do, which is incredible, no idea how this happens or why they do it, but they're just incredibly clever. To get out of becoming infertile or not being able to breed, they double the number of chromosomes in the cell. So instead of having one set from the mother and one set from the father, you have two sets from each. Woo! Peace out. So what happens then is if stuff from this doesn't match here, so you've got these, you've got one of this is a chromosome, and you've got one chromosome like this, and you go, oh no, they don't match. But fortunately, you've got clones, you've got two, so if it doesn't match here, this guy's going to match with this guy, and if this one doesn't match here, it's going to match with that one. So if you've got a mother one like, I shot my hand around, you've got a mother one like this, and a father one like this, and they don't match, the other one of these from the mother is going to come along and be here, and the other one of these from the father is just going to come along and be here, so they always match, and they can always be fertile because they've just cloned themselves so that when their chromosomes don't match with the opposite, with the other sex cell, they just take the chromosomes and match them with their own, which is really clever. So then the next generation will have 4 sets instead of 2, which is quadruploid or something. But it's called polyploidy because it's multiple ploids, and ploids are sets of chromosomes. And the fascinating thing is once you do that, you've got instant speciation. You've just created new species, just like that. Because it's different. It's got a different chromosome number, it's completely different. You've just made something completely new. And plants can do this in a single generation. They can create new species. And it's amazing. It's really common in plants. Really common. Polyploidy. Containing multiple sets of chromosomes, or more than 2 sets of chromosomes. And it's extremely common in plants. Between 70 and 80% of all plants have polyploidy, which means they are just always capable. They're always capable of hybridization and always capable of fertility. It's just genius. So then we talked about Spartina, which is a grass. It was a grass that was used. It grew on salt marshes in the south of England, particularly Southampton, just between Southampton and the Isle of Wight. But there's these big mudflats and salt marshes, and what we wanted was to be able to stabilise the land so they could be used agriculturally. But this plant couldn't do it. It wasn't strong enough to stabilise the mudflats on its own. So what we did was they imported the species, the same family, but they're different species. It's a very similar kind of grass from the USA. And they brought that in the hope that it would be strong enough to stabilise the marshes. And it wasn't. But you had these two different types of grass growing next to each other, and they hybridised. And the hybridised plant was very strong, and that's often the case with hybridisation, is that the offspring are incredibly strong. For example, we kept talking about horses and donkeys making mules, and how mules are just crazy strong, even though they're infertile, which is very useless. Damn mules, because they're so nice. It's just so pretty. Love mules. But they, so this hybridised offspring of these grasses was really strong, but it was completely sterile, so it didn't spread. It didn't, as with hybridisation, if it doesn't reproduce, it's almost useless at spreading. But because of polyploidy, one day, randomly, they were able to fertilise. Fertilisation was created, and then it just spread. It just spread like no man's business. So it went from being like contained in the Marsh to all the entire coast of the UK and Ireland and northwestern Europe. And it did exactly what they wanted it to do, which was to stabilise the marshes so that they could be grown on. And the fascinating thing was it didn't reproduce with either of the parent plants. In the end, this hybrid couldn't, it couldn't reproduce with the English grass or the American grass. And the fascinating thing about this was that it's a plant that's endemic to Western Europe, but it comes from speciation, from hybridisation with an Northern American species. So it doesn't grow. It's nothing to do with America, but it's also, you wouldn't find it anywhere. Completely endemic to Western Europe, but it's come from this hybridisation with American species. It's just very exciting. Everything's exciting. The thing about polyploidy was that you get infertile. It often produces fertile offspring. The whole point is that it enables fertilization, but it can produce infertile offspring in plants because if you have one plant that's got two sets of chromosomes and another that has one, then you end up with something that's triploid, and obviously triploid comes, then it's infertile. You can't have three. They wouldn't join up with anything. There'd be some, but there'd be a set on its own. Right. Okay, now we will move on to the longevity of species, which is really interesting as well. Everything is interesting. Just how quickly or lengthy species can form. And on average, it's the suggested estimation is that species last around 10 million years. And there are things that defy this completely. Obviously, as with everything, but based on fossil records and things, the suggestion is that species last around 10 million years. What we have got is things like the ginkgo tree. which is one of the longest lived species ever, as far as we know. And we found fossils of ginkgo leaves from the Jurassic period, which is just pretty cool. And the fascinating thing is it's almost unchanged. It's 120 million years, and it's almost unchanged. In fact, you could almost say it hasn't changed at all. The fossilized leaves that we've got are identical to the leaves that we find on the tree today. So this species has been around since before flowering plants even existed. They're just growing as well. I meant to go and look, and they didn't. But they've got some growing outside the university campus. It's insanely old. 220 million years. Flowers didn't even exist, and it's still around today. Now there's also the what the fuck have I written? Limulus, apparently. Limulus. King crab. It's Japanese. It's Southern Pacific. Northern Pacific. Mostly native to Japan. And that has, again, that's a species that, as far as we can tell from fossils and stuff, has stayed the same since the beginning of the Cretaceous period. So again, a hugely long period of time. Not as long as the ginkgo, but when discussing that, the point was made that it has a significantly shorter reproductive. What's what I'm trying to say? Basically, a tree is a tree. It lives for a really long time, and then its offspring will live for a really long time. Whereas, so there's less opportunity for variation to occur within a species or within a population. Whereas the king crab is slightly shorter than that because it is an animal that reproduces much more quickly at a much more rapid rate than the tree. Now then, the opposite of that, which is incredibly slow speciation, in fact almost not speciation at all, you have rapid speciation, or at least rapid differentiation, which is things like the Galapagos finches, which the Galapagos are really interesting because even though they're on the equator, they're in the middle of the onboard current, which makes them much colder. It takes Antarctica, I think it's Antarctica, and it carries it up, and it's significantly colder than it would otherwise be. And so the Galapagos climate is more temperate than tropical, and that's not what I said. It's less hot than it should be, is what I meant to say. But what happens is it has great big fluctuations in its climate, so you have periods of great drought. So whilst you have all these finches with their different beak shapes, what people found was in periods of drought, the small-seeded plants died first. So they've got the different beaks, as you know, to get into different seed heads. So the smaller beaks get in with the small seeds, seeded plants, and the ones with bigger beaks go for the stuff with bigger seeds. And but what happens is in drought is plants with small seeds die first, and the other ones stayed alive for longer in the drought. So during the drought, over single generations, so it's obviously birds don't have a very long lifespan, they're only around for about 18 months or so, the small seeded plants, because it was immediately beneficial during the period of drought for birds with bigger beaks to have bigger beaks. So, over single generations, there was a huge shift in the number of birds with larger beaks. And that was immediately beneficial, and therefore it was immediately brought into the evolution of the species, evolutionary genetics. And they knew that this was happening. It wasn't just maybe the beaks getting bigger. They actually found thousands of dead birds and as you know, you don't often see dead birds, even, if you think about how many birds there are, lots of birds, and how often they die, which is quite often because they have very long lifespans, but you only really rarely see a dead bird in general. So they found these thousands of dead birds, so they knew that they were dying because of the drought, because they couldn't eat in drought. So within each species and within each subspecies, each size grew bigger, but over just a few generations. And on the other side of the drought, all the birds had been ate, which is amazing. It's incredible. Then we moved on to talk about wallabies. Promise, I'm nearly done. I just want to tell you everything. So we have rock wallabies, which are, as marsupials, native to Australia. And it was rock wallabies that got captured and taken to Hawaii and kept in a zoo. And during the First World War, around 1916 or 17, these wallabies escaped, and they think there are only two of them. They're not entirely sure, but based on the records of the zoo and that kind of thing, they think there are only two, and they escaped, and they spread, and they were rediscovered in the 1970s, and people couldn't work out what they were. They were obviously wallabies, but wallabies are Australian. And people talked about where they might have come from, and they looked at the zoo records, and then they thought, oh, maybe there was a mistake when they were brought over, and that they aren't rock wallabies, they were something else. So people went to Australia and they searched all around Australia to try and find the wallabies that looked like the ones in Hawaii, and they couldn't find it. They couldn't find anything that looked the same. Because they had such differences in the ones in Hawaii to the rock wallabies in Australia. For example, their length. So an Australian wallaby is 1.21 metres long. This is all approximations, obviously. Oh, not approximations, but averages. And in Hawaii, the Hawaiian wallaby, they're about 1.03 metres long. The Australian wallaby's tail is 63.3 centimetres, and the Hawaiian wallaby's tail is only 51.6. That's 10 centimetres, more than 10 centimetres difference. It's quite a lot. The pelage, or fur length, in the Australian is 31 millimetres, and in Hawaiian it's 20 millimetres. So that's like a whole centimetre difference in fur length, which is pretty big. And in colour, they'd changed. So the Australian wallabies, rock wallabies, obviously brown, and the Hawaiian were pale grey, almost bordering on yellow. And they looked at blood, and they discovered there were significant differences in their blood proteins as well. And in the 70s, considering that this had come from two individuals, they thought there were about 100 of these rock wallabies, which is pretty incredible. And we're back. We've just arrived back. I'm going to have to go and fight in a second, but I'm going to finish this first. So anyway, the point was that they had to be inbreeding. So how on earth did they successfully manage to breed, given that they had such a limited gene pool to work from? Two individuals breeding over the course of 60 years, less than 60 years, they created a population of over 100. And how quickly they had changed, they had adapted to the climate of Hawaii. So understandably, all these things that made sense because going from a temperate climate like Australia to a tropical climate of Hawaii, and animals tend to be smaller, more tropical climates, having shorter fur, obviously because it's warmer, and the colour changing as well in terms of moving from a bush environment to a less bushy environment. And the thing is that if people hadn't known the origin, if we hadn't known that they'd come from this zoo and that they were rock wallabies from Australia, people really would have classified them as a separate species because they were so different, just so completely different. It's incredible. Amazing. I have no idea if they're still there, actually. I just feel like I should look this up and I have no idea. I'll do it in a second. Then what we did was we looked at evolutionary developmental biology, which is paleontology without fossils, which was really interesting, which is all about combining fossil knowledge with DNA. And the problem being that you can't get DNA out of those fossils, but you can get DNA out of things that are frozen in ice, and other bits and pieces. Basically, if it's frozen in ice, you can potentially get DNA out of it. If it's a fossil, you probably can't. It's fascinating. Yeah, this is the bit that I'm like looking at. I was listening to it, and I was like, oh God, if only Hank had then what we have now in terms of DNA sequencing, because he's just like guessing about genes, and the fact that people are talking about isolating genes in X-Men is kind of questionable in terms of when people were actually able. Though there was the video, which was the everything wrong with Days of Future Past, and the guy in that was like, why did it take them so long to get? They had Mystique's DNA in the 1960s. Why did it take them 50 years to get the Sentinels out and ready and that kind of stuff? It's like, well, actually, dude, you weren't able to actually put genes into other things until to extract specific genes and to isolate them and to put them into the DNA of other things until like 1995. So the fact that it took 50 years is kind of accurate, although all the same, there's like the fact that the Sentinels could actually adapt is kind of stupid because there would be some way of. Anyway, moving on to the whole thing. This is why I'm even trying to explain it. Anyway, the whole point is it used to, when they first took on the Human Genome Project in the early 1990s, and they did it, and it was incredible, but it took a huge amount of time, huge processing power in terms of computers, and it cost millions of pounds. Whereas now, it costs like £500 to £1,000 to sequence the DNA of something, and it takes about 24 hours. And people they have like these little things that are about the size of computer mice, like this, and it's got a USB, and you plug it into a computer, and you put your sample in it, and you go away, and it comes back in 24 hours with the entire DNA sequence is done. The problem is that DNA files are enormous, huge files, and the biggest bottleneck currently in understanding DNA sequences is the fact that we don't have enough computing power. It just takes so long, it takes so much computing power to be able to process the stuff. So the actual sequencing doesn't take very long, but the processing it afterwards into things that actually make sense and are understandable and we can work out what's going on takes a lot longer and a lot more power. So we'd have greater understanding if we had better computers, and obviously that's probably going to happen. So woohoo! Everything's looking bright. So you may remember, or whatever, that within a genome, within the DNA, it was commonly thought that there were genes and junk. So people could locate genes and they go, oh, this doesn't code for anything. It's just nothing. What is it? Oh, it's junk. What we know now is that junk DNA, though it doesn't code for proteins, it actually controls the expression of genes. For example, all of your cells have the same DNA, but the cells in your eyes don't produce the same proteins as the cells in your liver, for example. So the junk DNA actually determines what proteins are expressed, which is just what genes are expressed and what proteins are expressed. Which is really cool. I didn't know that, and it's fascinating. And so obviously this has to be the case. The incredible thing is that DNA is the universal genetic code. It's in everything, apart from very few examples that people have found that don't have the same genetic code. Everything else is the same. It's universal. DNA is the most ancient of evolutionary. It is of ancient evolutionary development. It is old. Obviously, it was in 1983 that Francis Crick proposed RNA world hypothesis, which was that RNA is the first ever data storage, basically, because it works as a protein, but it also stores data on making proteins. But RNA is very fragile, and it just breaks apart and gets reformed all the time. Whereas DNA is significantly more stable, so that's how it displaced RNA as the primary thing. Data. Way of storing data. Oh god. Okay, so a lot of evolution gets talked about how it's all about genes, but so much of it is not about genes, or is about genes. But it's not about which genes are expressed, it's about how and where they are expressed. For example, insects and other arthropods like spiders and even crustaceans have exactly the same genes. This is the guy that we had to change in lecture here, so this is... Who's this guy? I can't remember. Casper. This is Casper's stuff, but he's done this big study on insects and other arthropods. And the fact that insects and spiders, for example, have the same genes, exactly the same genes for legs, reducing limbs and stuff. But the leg number differs because of where the genes are expressed. So in a spider, the genes are expressed in more places, which is why it has 8 legs instead of 6. And the same thing, lobsters and other crustaceans, they have more legs because, yeah, so good. Now what you can use is comparative genomics. So basically understanding evolution by comparing genomes, but not just DNA, but also how it's used to express things. So genetic variation is ubiquitous. It would help if I could remember what ubiquitous meant, but you know. Basically, most genes are polymorphic, so you have different alleles, so they can be expressed differently. And genes and their regulatory mechanisms are the raw material for evolution, which we knew. Mutations lie at the basis of genetic change. This is true, but natural selection and random drift affect allele frequencies. So this is the theory that partially particular alleles of a gene are favoured because of natural selection. Here, it's more advantageous, but also this allele spread based on drift, and natural random drift is the assumption that a change in an allele is neither advantageous nor disadvantageous, and therefore it spreads randomly because it doesn't have any effect. So gene changes, so mutations can spread without being advantageous or non-advantageous. Yeah. So that's just a thing. It's not really leading anywhere, I don't think. It's just something interesting. So what we've got is comparative genomics, so molecular taxonomy. So this is so interesting. I think this is all I've written down. Okay, I'm just going to turn volume this bit. But this is a few interesting bits. Like it's not just about where genes are expressed, or which genes are expressed, but how long they're expressed for. And it was really interesting that actually human default is to have webbed fingers, a bit like mice and rats do, and that we don't have them because the gene is expressed for such a short amount of time. We don't have them. And with bats, for example, the reason that they have wings with their great big webbing is that it's a natural thing. It's the same gene that creates webbing in the feet of mice and such, but it's just expressed for such an extensive period of time that it turns into these entire wings, basically, which is amazing. And the fascinating thing, this is probably one of the coolest things I learned today, was that obviously lots of creatures develop through eggs, reproduce, and then you have the marsupials who reproduce with pouches and stuff, and then you have placental mammals. And the placenta actually comes, it didn't just develop, actually comes from retroviruses, in fact. What do retroviruses do? What do viruses do? They are parasitic, so they create a way to live off their host so that they can reproduce. And the genes for having a placenta come from a retrovirus. Because what the placenta do? It's parasitic. It draws nutrients, it draws energy, glucose, whatever, from its host. In the case of childbearing, it draws it from the mother, but the placenta gene didn't occur naturally. It was invasive. It was an invading gene that was transferred from a retrovirus into human DNA. Not DNA, but mammalian DNA. How cool is that? Microevolution changes in populations from generation to generation. Oh, I was just writing down notes. Microevolution and macroevolution. Macroevolution being broad scale, and microevolution being changes in the population's gene pool from just generation to generation. So, or big changes in the case of things like the finches, or the Hawaiian whatever it's called, rock wallaby. Yeah, I think that's it. But I hope you've enjoyed this video. I'm going to go and reply to your hangouts now because you're back like way sooner than I thought you would be, but it's quite exciting. Oh. Okay, bye.